Hello, Assalamu alaikum. The title of our presentation is Cellular Agriculture and Industrial Biotechnology for Food and Material. Our group presenters are Ravina, Ikraz Zunaira, and Sayyid Sibtul Hassan. Let's start our presentation with the introduction of this article. Transforming food system to provide a healthy diet and sustainable environment are the key goals of SDGs. Furthermore, even the food production of calories so far, but there is a still significant gap in providing sufficient food, and there are concrete problems related to low quality diets, which are micronutrient deficiencies and diet-related obesity. Moreover, the current estimate project for the food is requirement is 60% by 2050. In addition, domestication of livestock, adaptation of agriculture and industrial revolution causes the increase in the human population, which ultimately decreases the biomass on Earth. In conclusion, we need food alternative for food and material production. Next. So here we talk about the industrial biotechnology in agriculture. It is a key that to humanity they provide. It's a key to provide humanity with nutritious, safe, and healthy food, together with the chemical innovative material while reducing resource input to gain seasonal and geographic independence. Yeah. Next. Biotechnology is the form of many waves. That is red, green, white. Green biotechnology enables green revolution that include massive yield increase of crops through different breeding varieties, while red and white has been an cellular products. Cellular products are made of organic molecules like protein and fats and contain non-cellular or living material in the final product. Next. Cellular agriculture is the utilization of cell cultures of the whole variety of host organisms for the production of agricultural commodities rather than production by the farm, animal, or crops. In this article, we focus on cellular products that are non-genetically modified and focus on the production of cellular products, mostly in genetically modified organisms. As explained in the figure, that for the production of cellular and acellular products, we need host organisms that are animal and plant and microbes. Cellular products with examples are shown in the left, in vitro meat, and the acellular on the right, oval women. From now onward, my co-partners will explain further about host organism for cellular and acellular production of products. Thank you.
Assalamualaikum. My presentation part is about animal cells. As we know that how we can get our desirable products by using plants and microbial cells. Now with the advancement of biotechnology techniques in cellular agriculture, we can get many products just by using an animal cell. The main product that I am going to discuss right now is meat. Meat is an important dietary source of many components for supporting the growth, for development of human body, and for maintaining health. Now, through biotechnology, it is possible to get meat from an animal cell, and the meat that we get from an animal cell is called cell-derived meat or clean meat. So, clean meat is meat produced from cell culturing of animal cells. It is a prominent alternative for traditional meat that we derive from live animals. Next slide. There are many reasons behind it. First is that animal-based food products are considered inefficient as animals consume large amounts of food throughout their lives, of which up to 97% of calories are lost for processes regarding food, body maintenance and the production of non-edible tissues. Second reason is that animal agriculture is one of the major contributors to several environmental problems according to the report of Food and Agriculture Organization of United Nations. Animal agriculture is responsible for about 10% of greenhouse emission in the US alone, and globally for about 37% of all methane emissions and 8% of the freshwater pollution. Third reason is foodborne illnesses such as avian influenza due to the poor animal welfare conditions. Fourth reason is giving an excessive amount of antibiotics to farm animals, because the overuse of antibiotics induce the selection of antimicrobial resistant strains which is a major human health concern. It is estimated that by 2050, antimicrobial resistant strains will be responsible for more deaths than cancer. The fifth reason is that with an expected doubling of the global demand for meat by 2050, a traditional meat production system can't be considered sustainable. So, producing meat is in a sterile and controlled culture environment conditions could prevent these problems and not only improve food safety but also raising and slaughtering of animals can be avoided. Next slide. To produce cultured meat, stem cells are initially sampled from animals through a painless biopsy. These cells are then fed with nutrients in cultivators, where they multiply and differentiate. As they grow, they become muscle tissue, which is the main component of meat. A number of startups and the companies are currently working on developing a variety of cultured food from animal cells like beef, chicken, fish, seafood, milk, and cheese. Thank you. This is all from my side.
Assalamu alaikum. My name is Naira Naseem, and I will start by discussing the use of plant cells for cellular agriculture. Describing its environmental impact, first of all, the environmental impact of crop farming for human consumption is much lower than that of animal products. But still, that accounts for at least one-third of all agricultural impact due to the high production volumes and larger food loss and waste. Secondly, reducing the environmental footprint is an important factor to consider industrial biotechnology of plant cells for food applications. The worldwide adoption of plant stem cells, that is the de-differentiated plant cells as a source for cosmetic ingredients, is strongly driven by consumer demand for sustainably sourced ingredients. Cell culture technology was a prerequisite to access and exploit rare plants while securing supply without further endangering wild populations. Many cosmetic products are derived from cell culture extracts, but a number of formulations contain whole cells and therefore fall into the category of cellular agriculture. Now, discussing the use of plant cells as food, another impulsive for the utilization of plant cells as food originates from the nutritional recommendations to increase dietary intake of plant-based food. Firstly, there is a strong trend towards substitution of animal proteins with the plant-based alternatives such as soy and pulses. Secondly, there are many crops that contains the essential amino acids as compared to the animal-derived proteins. Thirdly, discussing the digestibility of crop proteins, there is a problem that many crop proteins, their digestibility is lower than that of animal proteins. But recently, some investigation shown that the nutritional composition of cultured plant cells revealed very favorable contents. And one of such content is that these samples exhibit differential digestibility, which is a basis for efficient absorption depending on species and processing. So considering that there is a strong trend for animal-based alternatives, also they contain essential amino acids and they have efficient absorption, these plant cells can be utilized as food. Discussing some other benefits of using the plant cell, first of all, there is the production of secondary metabolites. Plant cells can be used to provide the production of anti-cancer drug paclitaxel in 75,000 liter bioreactors. Also, there is an established practice to exploit tissue and organ cultures of ginseng for food supplement production. Secondly, plant cell culture medium. The plant cell culture medium is a chemically fully defined and consists mostly of inorganic ingredients, that is salt, sugar as carbon sources, and some low concentration vitamins and phytohormones. It is therefore that it is much less complex and costly as compared to the animal cell culture medium. The last benefit is that the plant cells could serve as material constituents, as 3D printing approaches could lead the way towards achieving ordered growth. It means that using this ordered growth pattern, plant cells can be utilized to produce a variety of materials because of their strength, etc. Thank you. That is all from my side. Now, Sayyid Sibtul Hassan will continue.
So basically, before concluding our presentation, we will talk about the microbial cells. So whole cells microbial production of food protein has long been established and commercialized. As we know that since this so-called the so-called single cell and mycoprotein must always undergo processing, downstream processing due to nutritionally unfavorable high contents of the messenger RNA. There are a number of foods that are being obtained through the process which involves microbes, but we are not going in detail of this. Next slide. So microbes basically can be cultivated to make synthetic materials. In nature, macroscopic materials grown from microbes such as biofilms, mushrooms, lichens have little relevance as synthetic structure material. However, microbes can be used to produce various polymers and polymer precursors for material. There are a number of aims that are associated with these techniques. One of the emerging field aim, is to use the microbial growth process as such for producing the synthetic material. Similarly, we have some challenges that need to be addressed. So one of the challenge is to control the growth and the morphogenesis of the cell to make tissues like material. The material fabrication using living organism is often termed as material biofabrication. And this technique is related to the biofabrication for tissue engineering and regenerative medicines. Next slide. So now I am going to discuss an interesting material example of fungal mycelium, and we will discuss the researchers' perspective about it. So an interesting material example is fungal mycelium, which can be grown into sheets or composite material. Filamentous fungi grow as long hyphae by tip extensions and branching and eventually form a familiar network. Interestingly, for centuries, humans have been using fruiting bodies of bracket fungi to make leather-like textile materials. Nowadays, designers and researchers are reviving this forgotten art and are able to control the mycelium growth into desired material and shape. In fact, the company Ecovative Designs has pioneered the use of mycelium as a material component. Fungal mycelium can be grown into bio-based solid composites, foams, and leather-like non-woven fabrics. Next slide. So now we will look towards the current opinions in the biotechnology. As you can see in the diagram, this diagram is representing a headset known as Corva, which is demonstrating the use of the microbially produced materials. The design demo is made from bioplastics, leather-like mycelium materials, synthetic spider silks, protein cellulose foams, and a composite of mycelium and bacterial cellulose. The headset was designed by Avian and the material was produced by VTT Technical Research Center of Finland Limited and Aalto University. Next slide. So before concluding our presentation, I would like to describe the applications of other microbes in the industrial biotechnology and the recent advancement. So in other microbes, we have seen that they have shown the capability to form materials such as bacterial cells of Bacillus species can be cultured to form biomineralized bricks in a microbially induced process. Microbial cells and virus particles can also be grown for biotemplating the synthesis of nanoparticles. For instance, gluconoacetobacter can be cultivated to produce very pure cellulose, non-woven films, and recently also 3D shapes. So it is an important to note that the described bottom up material fabrication process are guided by the genetic information of the growing cells. Thus, the understanding of the relevant gene factor will be highly important for the design of novel biosynthetic functional materials. Thank you.